Hello, and welcome to the second half of the Chapter 18 lecture on cell cycle. This is just a refresher of our objectives, and we are focusing today on number 3 and number 4. So, hopefully this is not the first time you've heard about the stages of mitosis, but we've got prophase. Some textbooks will include also prometaphase, metaphase, anaphase, and telophase. So the way I used to remember this was ip-mat, interphase, and then the mitosis, prophase, metaphase, anaphase, telophase. With this, it might have to be ip-mat in order to remember all of them. Whatever you need to do, right? So during mitosis, we've got our DNA that has been condensed and has been duplicated and lines up in the center of the cell. It gets pulled apart by this mitotic spindle that is formed of the cytoskeleton microtubules. Now, we've talked about how these chromosomes are formed, how the two sister chromatids are attached to each other, and we're going to learn a little bit more about in detail of each of these stages, but is specifically talking about separating the DNA first. So first thing, we know that the mitotic spindle is derived from centrosomes, right? And so these centrosomes, normally in the cell during interphase, there's only one. And so at some point, the centrosome has to be duplicated, and they have to get to opposite sides of the cell, and then they will kind of cast out kind of like a fishing line, the microtubule filaments to try and capture the chromosomes. And so during S phase, the chromosome will get replicated, centrosome will get replicated. And then during M phase, they will move to opposite sides of the cell. And so thinking again in this context of checkpoints and cyclids and CDKs, those CDKs that allow the cell to enter S phase, G1S, and the S CDKs are the ones that are responsible for also telling the centrosomes to be duplicated. Again, at mitosis, at M phase, they move to opposite sides and they start growing out these filaments of microtubules. And at this point, the structure is called like an aster. And if you've heard aster, typically people are talking about like a flower shape, right? So they're kind of making this flower shape. The microtubules you can see are coming out on all sides of the centrosome. And then eventually the centrosomes kind of figure out where they are. And the ones that are facing towards each other, those filaments are the ones that elongate. And they catch on to each other and form that spindle. So if you think of a spindle like what you would use to weave with, that's where the word comes from, because it kind of looks like a spindle for weaving fabric. So again, the centrosomes are formed by microtubules. Microtubules, if you recall, are dynamic. They are made up of little parts, and so they can be polymerizing or depolymerizing, and they're kind of randomly shooting out and coming back in again, growing in length and decreasing in length. And when they happen to become in contact with a microtubule from the opposite side of the cell, that connection is strengthened. And now you have an interpolar microtubule. It spans the entire length of the cell. And so now these centrosomes are often referred to as spindle poles, so poles, opposite sides. Now during prophase, we can see that the centrosomes are starting to form the mitotic spindle, right? This is the first stage of M phase. And another thing that happens during mitosis that might be easy to forget is that the nuclear envelope breaks down. So I'd like you to take a minute, pause the video if you need to. Why is it that the nuclear envelope needs to be degraded? So the mitotic spindle, those microtubules, they need to get access to the chromosomes. And so if the chromosomes are protected inside the nucleus, you see that the centrosomes are forming outside in the cytoplasm. It's a barrier that they can't get into. And so the nuclear envelope has to be degraded temporarily during M phase so that the centrosomes can grab onto the DNA and pull them to opposite sides of the cell. Again, that specific interaction, looking in detail how the microtubules attach to the DNA, is at these proteins called kinetochores. So the chromosome has a kind of a condensed region near the center that is called the centromere. And at the centromere, there are these proteins called kinetochores, and there's one on each side, on opposite facing sides of this replicated chromosome. So the microtubules will interact with the kinetochore and then pull the sister chromatids apart in opposite directions. And so this is going to create tension, like playing tug of war. And this tension is what allows the cell to line up the chromosomes in the center of the cell. And so there's actually feedback where these proteins, cytoskeletal proteins, motor proteins, are acting to tighten and to loosen the microtubules, to polymerize or depolymerize them. There's feedback that's going on, like to say, oh, it's loose on this side, let's tighten it up, or oh, we need to give a little on this side, so that the chromosomes can all be up in the center, which is pretty neat. And if you remember, middle metaphase, right? So where these chromosomes line up is called the metaphase plate. And this is a pretty cool picture. This is immunofluorescence or immunocytochemistry, in which they've labeled the microtubules in red and the chromosomes are labeled in green. And when red and green overlap in fluorescence, it makes yellow. And so you can see that the chromosomes are lined up at the center of the cell, and this plane where they are lined up is called the metaphase plate. Right? Then during anaphase, apart, the chromatids separate. And here you can see nicely the mitotic spindle is in red, the chromosomes are in blue. You can see that in metaphase, they were all lined up. During anaphase, they get pulled apart and kind of looks like a monster's gaping mouth. Right? Now, how does this actually happen? How do those sister chromatids detach from each other at anaphase? So you're going to learn a little bit more of the molecular signaling that involves this. So if you recall, when we talked about chromatids and chromosomes, how they're associated with proteins, the mitotic chromosomes, they are condensed and you have two sister chromatids that are interacting at the centrosome region. And so you can see that they have these proteins called cohesins. And the cohesins are the proteins that help hold those two sister chromatids together. Now, during anaphase, those cohesins need to be degraded. And so there is an enzyme called separase, pretty creative, right? And the enzyme separase cleaves those cohesin proteins, and that allows, when there's this pulling going on by the microtubule, that the sister chromatids will finally let go and be pulled to opposite sides of the cell. So separase is normally kept inactive by an inhibitory protein called securin. And so something must turn separase on. There must be a signal that tells separase, all right, the chromosomes are lined up at the metaphase plate, we're ready to move on to anaphase. And what do you know? We have something that looks a little familiar, hopefully. We have APC, anaphase promoting complex. And so active anaphase promoting complex will come in and break down that inhibitory protein securin. Now separase is active, it can break apart the cohesins and allow anaphase to occur. Hence, anaphase promoting complex. Now, a little bit more detail of what happens during anaphase that might be new. So these chromosomes are getting pulled apart from each other, and there's two ways that this happens. The two methods are called anaphase A and B. These are both happening at the same time. So first of all, you could have the microtubules that are attached to the chromosomes be shortening, right? They could be depolymerizing. And so they're just getting shorter in length, and that's what's pulling the chromosomes apart. However, you can also have the poles pulling apart from each other in distance. And so even if the microtubules aren't changing in length, the poles they're attached to are going further and further away from each other. Oh, there's Rocket. So after anaphase, right, what happens next? So in order to for mitosis to be completed, right, we said that we have this checkpoint, right? For anaphase to actually occur, we have to have this checkpoint passed in which the cell recognizes that all the chromosomes are lined up at the mitotic on the mitotic spindle. And until those chromosomes are in that state, the cell will not go on past this checkpoint. When chromosomes are not attached to the microtubules, they actually send a stop signal to the cell cycle control system. So once all the chromosomes get attached to the mitotic spindle, then finally the OK signal is sent out. And again, how does this happen? So if a chromosome is not attached to the microtubules and not lined up at the center of the cell, it will create a signal that blocks activation of APC. If you remember, right, anaphase promoting complex. So for the cell to leave metaphase and enter into anaphase, it needs to have this active APC enzyme. And so if the microtubules aren't attached properly to the chromosomes, this APC protein will not be active. And without APC, if you recall, the sister chromatids cannot separate. So here's just showing that figure again, right? If the chromosomes are not lined up at the metaphase plate, they will not allow APC to be activated. Once APC does get activated, it can turn on separase, which will allow the sister chromatids to get pulled apart. And the way it does that is by breaking down those cohesin proteins. After anaphase, we have telophase. The DNA has been separated to opposite sides of the cell, and so we don't need the mitotic spindle anymore, so that will disassemble. And the nuclear envelope is going to reform. This time there's going to be two sets, right? One around each set of DNA on opposite sides of the cell. And the DNA does not need to be compact anymore because they're not organizing DNA, and so it can start to relax. So the way that the nuclear envelope is disassembled and reassembled is kind of interesting. So let's look in detail first at the way the nuclear envelope is structured. You can see in this example on the top of our circle, here we have a nuclear envelope. We've got the phospholipid bilayer, and there's actually an outer and an inner phospholipid bilayer. So there's two bilayers in the nuclear envelope. And then underlying this is lamin protein. And this helps to promote the structure and the shape of the nucleus. And then, of course, we have our DNA in here. We also have some nuclear pores, some transmembrane proteins that allow things to enter and leave the nucleus. So when the cell gets a signal that it is time to degrade the nuclear envelope during prophase, what happens is things get phosphorylated. So the nuclear pore proteins as well as the lamins get phosphorylated, and the addition of this phosphate group will cause them to become destable and to separate into little pieces. And so now the nuclear envelope is gone. And then during telophase, the nuclear envelope needs to reform. And so, just as phosphorylation causes the nuclear envelope to break apart, by dephosphorylating or removing those phosphate groups from the pore proteins and from the lamins, that's going to allow them to start to aggregate together. And they aggregate into these little vesicles that eventually fuse together and to reform an entire nuclear envelope. Now, after mitosis, we have cytokinesis, cytoplasm, right? So breaking apart the rest of the cell. So the DNA has already been separated, but the rest of the cell needs to be pulled apart. And this looks pretty neat, right? There's a contractile ring that forms. It's kind of like if I was to put my hair in a ponytail with a rubber band, right? And pretty interesting. So we talked about that metaphase plate, the location where the chromosomes line up in the cell. So where those chromosomes line up will tell the cell later where to cleave the cell in half. So we have a contractile ring is made out of actin, so it's really important to keep track. The mitotic spindle is made out of what? Microtubules, whereas the contractile ring during cytokinesis is made out of actin. And then there's also myosin proteins that help to tighten that ring. And again, this is just on the underside of the cell membrane, right? And so here we have some electron micrograph images of cells dividing. And you can see, especially in this lower image in B, you can see in the center, right here, this dark, dense, electron dense region, that is actin that you're seeing and myosin, right? That's contracting that. And you can also see these elongated lines. These are remaining microtubules from the mitotic spindle, which is pretty cool. Now again, as I said, where the mitotic spindle lines up, where the microchromosomes line up in the cell during metaphase determines where the cell will cleave in half during cytokinesis. And so we always draw the metaphase plate right in the center of the cell, and that will result in the cytokinesis occurring right in the middle of the cell, and you get two daughter cells that are even in size. But that does not always happen. So we draw it in the center for simplicity, but sometimes the metaphase plate may be off-center in one direction or the other. And when that happens, also where the cell divides will be shifted off of center. And what's going to happen is you're going to have one daughter cell that's larger than the other. And that's okay. For example, when the two offspring are going to end up being different cell types, you want to have different molecules inherited in those daughter cells. So remember, we've got all sorts of molecules and proteins floating around in the cytoplasm, and it's just depending on where they happen to be when the cell cuts in two, that determines which molecules and what concentration of these molecules are in the two daughter cells. And so you may need different proportions of molecules in order for one daughter cell to become one type and the other daughter cell to become something else. Again, this contractile ring, it's made out of cytoskeletal filaments, specifically actin, and then myosin is a motor protein that helps to contract those fibers. And again, this is just right under the surface of the cell membrane. Here's a picture showing a cell that's dividing, just looking under the microscope. And you've looked at cells now, so this probably looks kind of familiar. You can see that during interphase, when the cell's not actively dividing, it's kind of spread out and flattened. And then during cell division, it rounds up. Remember we talked about and you saw when cells detach from the surface of the dish, they get really round and shiny. And you can see that in this picture here. So if you see cells that are round and shiny like that, they could be in the middle of division, cell division, or they could be dying and detaching from the plate. But here we can see kind of nicely the anaphase stage and then the cytokinesis. And then the last thing I want to note is look at these two daughter cells. Do you see how they are mirror images of each other? So this is quite often the case in terms of the shape of the cells of the two daughter cells, which is pretty cool. Now you may say, or maybe you've never thought about it, what about the organelles? right? We know how DNA gets separated. We learn that over and over through high school and college classes, but what about the rest of the organelles, the mitochondria, the endoplasmic reticulum, right? Well, you're going to find out today. So the mitochondria and the chloroplasts, basically they get duplicated when the cell is getting ready to divide. So there's double the amount, and then they just get split evenly among the two daughter cells. The endoplasmic reticulum is a bit different. If you recall, it is actually continuous with the nuclear membrane. And so during M phase, the ER is released from the nucleus, right? So the nuclear membrane normally is continuous with the endoplasmic reticulum, but that connection gets cut off when the cell is going to undergo mitosis. And then the ER gets cut in two during cytokinesis. The Golgi apparatus will fragment into little vesicles, and then those pieces will actually attach to the mitotic spindle. So we don't see them in our little cartoons, but in addition to chromosomes being attached to the mitotic spindle, we also have Golgi apparatus. And so the mitotic spindle helps to pull apart pieces of the Golgi as well. And then everything else is inherited randomly. So, there's stochastic movement in the cytosol. Wherever those organelles, ribosomes, proteins, molecules are in the cytoplasm is where they will end up after the cell divides. Now let's talk about cell number and size. When we talk about cell growth, you may be thinking, okay, is she talking about the size of the cell, or is she talking about like the number of cells, right? So we could be talking about either, right? So for right now, let's just talk about cell size. Now, if you look at a human or a mouse fertilized egg, an embryo, right at the very beginning after fertilization, these are actually equal in size. But you know that an adult mouse and an adult human are not the same, right? And similarly, our organs have different sizes depending on species. And so scientists are still learning about what signals tell these organs what size they should become and what size these cells should be. There are some things that we do know. So there are programs in the cell that regulate growth, so the size of the cell, division, cell proliferation, and death. And so the balance between these three things is what is going to determine how big an organ is, right? So let's talk about cell death in more detail. So purposeful cell death is called apoptosis, otherwise known as programmed cell death. And so again, the tissue size, one aspect of it is a balance between cell death and proliferation. If cells are dying and being born at the same rate, the organ will stay the same size, right? If apoptosis increases compared to proliferation, now that organ will shrink. And if proliferation is ramped up and apoptosis does not change, then that organ will grow in size. Apoptosis is not always bad, right? Especially during development, apoptosis is actually really important. Such as during development of the brain, we actually make way more neurons than we need. The bone marrow and intestinal cells, these are turning over, and so there is a lot of apoptosis occurring in those tissues. So again, I said that we have more neurons are made in our brain than ultimately are needed. And so basically kind of sculpting or paring down of this organ is needed. And this also happens in other species and other organs outside the brain, such as finger formation, right? So originally we have more kind of like mitts, and then the cells in between where the fingers are going to be undergo apoptosis to allow us to have are separate individual fingers. And the same thing happens during development in frogs when the tadpole needs to lose its tail. That is by apoptosis. So apoptosis is not always bad. Now, again, we have to have this balance of apoptosis and proliferation, and some tissues are able to proliferate and regenerate better than others. Such as liver, you can provide a liver transplant to someone. A piece of your liver would be removed, not the entire liver, and that piece that gets removed can actually grow back. Right? So that liver somehow knows that the tissue's been lost and will upregulate cell proliferation in order to regrow that tissue. And scientists have started to figure out the mechanisms behind that, and they can actually stimulate liver growth with a drug called phenobarbital, which is kind of neat. Now, the opposite of apoptosis is necrosis. So this is usually caused by disease or damage, like mechanical damage to tissue. And this is messy. It can affect other cells nearby. So necrosis can induce inflammation. It can release toxic molecules to surroundings and can kill cells around it, whereas apoptosis is a much cleaner method of cell death. And so here you can see on the left we have a cell undergoing necrosis, and it's just kind of like, right? And then in B and C, you can see various stages of a cell undergoing apoptosis. So we're going to talk about what these little blobs are inside of that cell in B. And then what I'd like to note about C is that the cell, when it undergoes apoptosis, it can actually be eaten by immune cells, like a phagocytic cell is surrounding and has engulfed this entire dead cell that underwent apoptosis. So some features of apoptosis. We have blebs, so we saw those in the previous picture. This is kind of bulges of the cell. The cell will shrink in size and condense. It's in the materials inside of it. The cytoskeleton breaks down, right? That would contribute to the cell shrinking in size. The nuclear envelope disassembles, and the DNA actually fragments, breaks apart in pieces. In addition, the cell surface is changed to recruit these phagocytic cells. If you remember back when we learned about the cell membrane, we learned about phospholipids, right? And we just learned about phospholipids in general, their general structure, but there are different varieties of phospholipids, phosphatidylcholine, phosphatidylserine. And we also talked about how the arrangement, whether these phospholipids are on the outer layer of the bilayer or on the inner layer of the bilayer, can be regulated by flipases and scramblases, right? So during apoptosis, those enzymes get activated and they switch phosphatidylserines, the specific type of phospholipid, from one layer to the other, and that acts as a signal telling surrounding cells, hey, this cell is dying and it needs to get eaten, which is pretty cool. Now, what's going on inside the cell? What signaling pathway is telling the cell to undergo apoptosis? The key enzymes for signaling of apoptosis are called caspases. Right. So in this picture on the right, we have two enzymes here in brown called procaspases. So just like a precursor mRNA or precursor protein, these procaspases, they're not yet active. There's a piece of the protein that has to get cleaved off, broken off, before this enzyme is active. And so normally these procaspases are just hanging around. They're not doing anything, not inducing cell death. But when a signal arrives to tell this cell, or from within the cell, that it needs to undergo apoptosis, these procaspases will get cleaved. And as I said, that cleavage will activate them. These two pro-caspases will bind to each other to form one active caspase molecule. And what caspases do is they cleave proteins. And so you can see that this we have this cascade occurring, right? So one active caspase can not only cleave just one target, but many. So it will cut apart one protein, move on to another, cut apart that protein, and keep going. And then each of those, the targets that this caspase is cleaving, are additional caspases. And so you have caspases, cleaving caspases, cleaving caspases, kind of like when we had our MAP kinase, kinase. Right? So we call this an amplifying cascade, caspase cascade, and that is because all of the proteins involved are called caspases, and this signal is getting amplified. See how we had one initial caspase called an initiator caspase? Because it can target more than one protein, this is going to amplify that signal, and because there's multiple levels of signaling, it's called a cascade. And now, ultimately, these executioner caspases will be turned on, and their targets are going to be things like cytosolic protein and nuclear lamin, right? We talked about how during apoptosis, the nuclear envelope breaks down. Just as a reminder, how are all of these caspases getting activated? Just like this initial example, they get activated by cleavage, which is very nicely done by other caspases. So again, we have initiator caspases. They start out as pro-caspases, and only once they're cleaved do they become active. And then they induce this amplifying caspase cascade that leads to the cleavage and activation of executioner caspases. Now, as you can guess, since apoptosis, you can't really revive cells once they're dead yet, it must be very tightly controlled. And so there are layers of regulation around apoptosis. So what starts this cascade? There's a family of proteins called the BCL2 family. And these are the proteins that activate these caspases. So again, these pro-caspases are just hanging around at constant levels. It's just that they are not yet active unless they get cleaved. And so within this BCL2 family, there's a variety of proteins. Some of them act as inhibitors, so whenever they're present, they are keeping the cell from undergoing apoptosis. And others, when they are present, they will promote apoptosis. Two examples of proteins in this family that induce apoptosis are called BAX and BAC. And so these ones in particular, if there is a lot of DNA damage and the cell needs to undergo cell death, these proteins are ones that can get induced. And the way this works is that they release a molecule called cytochrome C. In this picture, we actually have the initiation of apoptosis occurring in the mitochondria. So mitochondria store cytochrome C molecules. They keep them out of the cytosol. And when the cell receives a signal to undergo apoptosis, you can see that BAX and BAC get activated. And what they do is they go to the mitochondria and they allow cytochrome C to be released. And now we have cytochrome C free in the cytoplasm. And it will interact with an adapter protein and form this kind of wheel-like structure. And then they will grab onto or recruit an initiator caspase, in this case called procaspase 9. And this forms a complex called the apoptosome, or wheel of death. all right? And so these proteins within that apoptosome, the procaspase will get cleaved and activated, and then these initiator caspases can go down and activate, induce a cascade and activate executioner caspases. So there are extracellular signals that can tell a cell to undergo death. They can also receive signals from inside the cell, right, like DNA damage. And so we talked about how DNA damage, that's an interior problem inside the cell that can tell the cell to undergo apoptosis via BAX or BAC, and that those are pro-apoptosis proteins. However, if a cell receives a signal from the outside, from its surroundings telling it to undergo cell death, this target is going to be BCL, a different protein in the BCL2 family. So when an extracellular signal is telling the cell to undergo apoptosis, it needs to bind to a receptor, and these are called death receptors. And one example is called the FAS receptor. Here we have a cell, the larger cell on the bottom here is a cell that's going to be told to undergo cell death. And you can see that it has this FAS death receptor on its surface. Now here it's going to receive a signal to die by a killer lymphocyte. And so just as there is a death receptor, there is a ligand for it. And in this case, the ligand is actually attached to the surface of this killer cell. So when this killer lymphocyte gets close enough in contact to bind the death receptor, it will initiate the formation of this complex called the DISC complex. And ultimately, it will cause activation of a caspase called caspase 8. Now, I don't require you to remember like caspase 8 versus caspase 9, but I do expect you to know the difference between initiator and executioner caspases. And in terms of this pathway here that we're looking at, I would expect you to know what a FAS death receptor and ligand are, and that a DISC complex form, and then it leads to activation of an initiator caspase, right? In addition to telling cells to die, there's also extracellular signals that tell them to divide or to grow in size. So there are survival factors from the environment that tell the cell not to undergo apoptosis. We've talked about mitogens before. These will tell cells to divide. There are growth factors that tell the cell to grow in size, as the name implies. And these are not mutually exclusive. So you can have a protein that is both a mitogen and a growth factor, or a mitogen and a survival factor. So again, these survival factors are keeping the cell from undergoing apoptosis. And so I want to look at an example of this in brain development. If you remember, I said that we are born with more neurons than we need, and that our brain needs to be kind of sculpted and pared down. And the way this works is by the use or absence of survival factors. So if a cell does not receive survival factors, it could undergo apoptosis. And that's exactly what happens here. You can see that during development, we have a lot of nerves that are born, and then they have to connect to something, right? Nerves have to synapse with target cells. And so in this case, their target cells will release the survival factor. And so if a neuron is close enough to its target cell, it will receive that survival factor and it will be able to survive. However, if a cell is born and it's not in contact with the target cell, if say there's too many neurons, that neuron will not receive the survival factor and it will undergo apoptosis. Right, so this helps to match the number of nerves to their targets. Right, so again, a survival factor, if it's present, it will prevent apoptosis. Now let's look at this more in detail of the signaling pathway. Here we have a survival factor, and somehow it needs to tell apoptosis not to occur. And so if you remember, what proteins are involved in apoptotic signaling? It's that BCL2 family. And we said that the BCL2 family contains some proteins that are pro-apoptotic and others that prevent apoptosis. So these survival factors are going to be allowing the induction of these proteins that are inhibiting apoptosis. You can see a survival factor is binding the surface of a cell at a receptor and leading a signaling cascade, which ultimately activates a specific transcription factor for this BCL2 gene that codes for a protein in that family that prevents apoptosis. So it's kind of nice. You're starting to see all the things that we've been learning are coming together. Again, we've talked about mitogens before. These are going to signal a cell when it is in G1 phase to go into S phase. And again, just like any other extracellular signal, they're going to bind to a receptor and they are going to induce a signaling cascade in the cell. And for a cell to go from G1 to S, we've learned about checkpoint proteins, right? And so we learned that in order for a cell to enter into S phase, this RB protein needs to be inactivated. This is one of those checkpoint proteins, a regulator. We have G1 and G1S CDK that are, when they become active, it allows the cell to leave G1 and to enter S phase. And so the way this works, as we've learned before, an extracellular signal will bind outside the cell to a receptor that will cause a signaling pathway that will ultimately activate these CDKs, right, that are important for procession into S phase. And the way the cell is able to actually move on into S phase is by inactivation of RB protein. Now, RB protein normally will bind to transcription factors that are important for producing genes related to cellular division. And so the kinases, CDKs, they phosphorylate things, their target is RB. And so when RB gets phosphorylated, it inactivates, it forces it to let go of that transcription factor, and now these genes required for cell proliferation and cell DNA duplication can be expressed. Let's look at another example, platelet-derived growth factor, or PDGF. So normally, if there's an injury to the body, it's going to cause cell death by necrosis, and there's also going to be blood clot formation. Now, these blood clots will release PDGF, and what this is ultimately going to do is it's going to start cell proliferation for this tissue to heal itself. Right? So the blood platelets in this clot release this growth factor, hence why it's called platelet-derived growth factor. And it will bind to a receptor tyrosine kinase. Does that sound familiar? Hopefully it does. This will initiate an intracellular signaling cascade that will ultimately result in the cells at that site of injury undergoing division to help replace lost cells and to heal that wound. And there's a similar thing that happens in the liver, a hepatocyte growth factor that will also induce proliferation. We talked about how you could donate part of your liver. Do not memorize this, but I just want to show you because now you can understand what's going on, which is pretty cool. This is a pretty complex figure. You can see that a mitogen, such as PDGF, is binding to the surface of the cell at its receptor, and it is going to be activating these tyrosine kinase receptors. What do? They autophosphorylate each other. They phosphorylate target proteins that form a scaffold right near the surface of the cell. And we've learned about RAS proteins, right? These are GTPases. When they get activated, they are ultimately going to activate this downstream signaling pathway that ultimately is going to activate some MAP kinases. In this case, these ones are called ERK, and they are going to lead to changes in gene expression. So, what genes are being targeted? If we want the cell to undergo proliferation, it needs to leave G1 and enter S phase. So the end target of this signaling cascade are these genes. And if you remember, we talked about RB. RB is important. It's preventing these genes from being expressed. It's getting activated, and that transcription factor, which happens to be called E2F, can now be released and bind to these genes promoters and activate those genes to be expressed and then to be made into proteins. That will cause those cyclins to rise, which will then activate those SCDKs and allow the cell to enter into S phase and proceed on with cell division. Now again, growth factors are important for the size of the cell. We've talked about how a cell, every time it divides, it splits in two. Now it's half the size. And then when those cells split in two, if they don't get enough time to regrow, then they will also be half their size. And so if you don't allow time for cells to grow between cell divisions, the size of the cell will just shrink and shrink and shrink. All right. So, excuse me, sorry. There are cells that become terminally differentiated. In other words, at a certain point they stop dividing, they mature, and then they just do their functions and they never divide again, such as neurons, right? And so a lot of times it makes sense, right? You don't want to invest a ton of time growing really complicated shapes or structures or really large size if you're just going to divide again. And for a cell to grow in size, think about what needs to happen. You need to have a lot of protein being made. And so growth factors are going to ultimately cause increased protein synthesis and also reduced degradation, right? We know that ways to regulate the amounts of protein in the cell are either by increasing the rate at which they get made or reducing the rate at which they get broken down. And so again, these are extracellular signals. They're going to cause signaling cascades inside the cell that are going to increase protein synthesis and decrease protein degradation. And so depending on the cell type, you can have some cells, such as neurons. This is a terminally differentiated cell that has grown quite large in size versus this other cell type that is not as large. Now, we do have some signals that are inhibitory, such as myostatin. And so this is just kind of a cool example that I wanted to show. So myostatin inhibits cell growth and proliferation. So this is an extracellular signal that if the cell receives it, it's going to stop growing in size and not divide as much. So these mad scientists wonder, what happens if we inactivate myostatin? Right? If you do this, this is specifically related to muscle cells. It's going to cause these muscle cells to grow in size and also in number. So this tissue size is going to grow really dramatically. And so that is how you get this very fit cow in this picture. And the most swole mouse I have ever seen. It's quite disturbing looking. But pretty interesting. So all they did in these two species was they inhibited the synthesis of this myostatin signal. And so again, the cells in the muscles grew larger in size and the cell division rates increased. Pretty neat. Hope you enjoyed this chapter. And we only have 1 week left. So best of luck.